Hello, everybody. This is Chapter 3 in our textbook on cells from the human body and health and illness. And what we're going to look at today is the smallest unit of living matter. Okay, uh, these are the topics we're going to cover. We're going to look at a diagram of the parts of a typical cell and label each of the components and indicate what they do. We're going to talk about cell transport, that is how cells bring stuff in or kick stuff out of their interior. We'll talk about the cell cycle and mitosis. We'll talk about differentiation, and we'll talk about cell growth and apoptosis, and a little bit about cancer as well. So cells are the smallest unit of living matter because they can do everything that we think of as a life process. They have the ability to grow. They have the ability to move. They have the ability to respond to their environment by changing their actions or um, their characteristics. They have the ability to reproduce. They have the ability to take in fuel and metabolize it and produce waste. They have organization, and they have boundaries. And all of those are hallmarks of what we consider to be a living thing. Okay? So this is why a biologist would say that we call this the smallest unit of living matter. Typical cell is shown here. Okay? First thing you see when you approach a typical cell is the outside, and that's the cell membrane. Okay? Now the cell membrane is basically to keep the interior of the cell in one place, right? So it's like a fence around a gated community, right? It makes sure that the material the cell needs to survive doesn't go wandering off into space so that it's no longer useful. It also has the ability to allow only certain things to pass through it. Okay, so it has a gatekeeping function where the cell can figure out whether it wants to bring a certain component in or whether it wants to kick something out. So the cell is able to um, control the membrane in terms of how permeable it is and what sort of material is allowed through it. It also has the ability to tell the cell about the outside environment. Okay, there are special molecules embedded in the membrane that are called receptors that essentially monitor the outside, and when they pick up a particular change, they can relay that information to the inside of the cell, and the cell can respond accordingly. Um, these these changes that it produces in the cell are generally the result of chemical reactions. Okay. Um, You'll also notice in the cell that you've got these little chambers all over the place. These are called organelles. This is a mitochondria. This is the nucleus. This is the Golgi. This out here is the rough endoplasmic reticulum. This is the smooth endoplasmic reticulum. You've got lysosomes. You've got free ribosomes. And you've got cytoskeletal components like microfilaments and microtubules and intermediate filaments, and then centrioles inside of centrosomes and secretory vesicles as well. Okay, so what do all these do? Well, we'll get into the particulars as we move through it, but just to give you a sort of an overview, okay, the nucleus happens to be where the information that the cell needs to survive is stored. It's stored in a molecule called DNA, and the pieces of information are on this long DNA molecule in little packets that are called genes. And what the genes contain are the instructions necessary to build a particular protein. The nuclear membrane sort of holds all that material in one place, and there's openings in it so that stuff can get in and out through the nuclear membrane. Outside the nuclear membrane is the rough endoplasmic reticulum. That's the aquamarine stuff with the little red spots on it. And that's where proteins get made that are going to end up either being inserted into the membrane or secreted by the cell into the outside. or taken up by one of these other chambers so that those little, we call them organelles in the cell, can do their job properly. Okay, so the little red ribosomes are the protein builders. The secretory vesicles and the transport vesicles are little bubbles made of the same material as the cell membrane, and they are capable of moving around inside the cell, and you can see some examples of them here. These are transport or secretory vesicles, and they shuttle stuff between different locations. There are transport vesicles that are going to move stuff between the rough ER and the Golgi, and secretory vesicles that are going to move stuff from the Golgi to the cell membrane or to another organelle. Okay, and they all use energy to do that, and they kind of walk around on these cytoskeletal components like little train tracks. Okay, these little things that look like wieners are where the cell gets most of its energy in the form of something called ATP, and that allows the cell to do work. Okay, so we call these the powerhouses of the cell. These are the mitochondria. Lysosomes are there to essentially break down and recycle anything that is worn out and needs to have its parts reused and then other parts thrown away. Okay, so there's stuff inside here that's very, very destructive and needs to be contained inside this little chamber. When the cell decides that it's going to end up committing suicide, one of the things that happens is that these little chambers rupture and the cell chews itself up from the inside out, and the cell does no longer function. And then the smooth endoplasmic reticulum out here is where the cell stores a lot of calcium and does detoxification reactions, and also manufactures things like steroids and fatty acids, and we'll get into what those are in a bit. Okay, so we're going to go through each of these piece by piece in more detail and learn what each of these components do. So here's a look at the cell. So as the cell membrane indicated, it covers the outside of the cell, and it determines what goes in and out, and it has the ability to admit certain things and other things it does not admit. Okay, the middle of the membrane here, all right, is made up of material that doesn't mix very well with stuff like water or stuff that dissolves in water. Okay, these are fatty acid tails, and we would say that they are hydrophobic, which means that they are water fearing. So they don't like to mix with water. They like to be with their own kind. And because of that, this is a barrier to a lot of material that would like to cross the cell that either has an electrical charge or that dissolves very easily in water, even though it doesn't have an electrical charge. And so how does that stuff get through the membrane if the cell needs it? Through little tunnels, gates, or pumps that the cell makes and puts in the membrane, and the cell can control how many of them there are and when they're open and when they're shut. Okay, there are some things that can go right through the cell membrane, and those tend to be things that are what we would call hydrophobic or water fearing. Okay, examples include oxygen molecules and carbon dioxide molecules and other stuff like cholesterol and steroids. Okay, that slips right through without any special tunnel or gate or pump. And also water can get through one of two ways. It can either go through the membrane on its own because it's tiny, or it can go through a special tunnel called an aquaporin and go from one side of the cell to the other. The nucleus is where the information that the cell uses to exist is located. Okay, so it contains a long molecule called DNA. All right, and in a human cell, there's a total of 46 of these DNA molecules, and they're called chromosomes. And these molecules are usually attached to proteins that are called histones, and so that combination is called chromatin. Okay, The nucleus is surrounded by a double-layered membrane that has pores in it that let certain things in and out. The nucleoplasm is what the chromatin is suspended in. The nucleolus is a region inside the nucleus where ribosomes get made, and of course those are the little particles that build protein for the cell. And the chromatin, as we already talked about, is just the DNA and the histones, and the information blocks that contain the protein building information are called genes. Okay, so each gene is a recipe for a particular protein, and the proteins that the cell uses help the cell to survive. Okay, proteins do all the work of life. So they do things like speed up chemical reactions and defend the cell against attack and provide the cell a framework so that it has shape, and also allow certain things to move from one place to another using ATP for fuel. Okay, and they also serve as receptors, which tell the cell what's going on outside, and then they also can serve as gates and pumps and tunnels. Okay, those are just few of the roles of proteins. And so occasionally the cell needs a particular protein made, and what it'll do is it'll find that information in the nucleus and then make a copy of that information and bring it out into the area between the nucleus and the membrane, and then the ribosomes will use that copy of the information to build the protein that the cell needs. Okay, we'll get into the particulars of what's involved in a little bit. The cytoplasm is just a gel-like substance that's inside the cell, but it's more complicated than just a gel. It has waste products and nutrients and stuff like oxygen and carbon dioxide in it. It's got water. Okay, it has a lot of proteins that are suspended in it, like the microtubules and the intermediate filaments, and the microfilaments are in there too. And then the organelles are in there, and those are little chambers inside the cell, or little particles, where certain chemical reactions that help the cell survive happen. Okay, and the reason that the cell uses organelles to do this is so that the chemical reactions are more effective, because everything's in one area, and it also separates chemical reactions that would get in each other's way. Okay, so it keeps a set of destructive chemical reactions in one location and a set of chemical reactions that builds things in a different location, right? And that way the cell can do both things and not waste its time. Okay? So what are some of the organelles? Well, one example is the mitochondria. Okay, it looks like a little wiener. All right, it's about the size of a bacterial cell, and it has two membranes around it, an outer and an inner membrane. And the inner membrane has a lot of ridges and grooves in it, and that's designed to increase the area available to do work. And the kind of work that the mitochondria does is it makes ATP. And ATP is a form of chemical energy that the cell can use right away to do some sort of a task, okay, whether it's to move or to build something. Okay, the energy to make ATP comes from the fuel that the cell takes in from the outside through its membrane, and the cell is able, through a series of chemical reactions, to steal the energy from that fuel and to send it over to ATP, and then they use the ATP to maintain the cell as a living thing. Okay, it's a little bit like the gas in your car, right? You know that when you put gas in your car and then you put your key in the ignition and turn it and step on the accelerator pedal, that your car will move. Okay, and that's because your engine and your carburetor understand gasoline. Okay, but gasoline comes from crude oil, and you can't put crude oil in your gas tank because your engine and your carburetor won't understand it. So what has to happen is that the crude oil has to be broken down into smaller pieces. The one that's in gasoline is an 8-carbon piece called octane, and that's what's in gasoline that your engine understands, that allows your engine to run and your car to move. Okay, so the same thing happens with fuel for the cell, right? The cell steals the fuel from the molecules it takes in for energy, and it hands them over to ATP, and then the ATP is used to do the work. And then in that process, there is usually going to be some heat generated. All right, we need ATP in order for chemical reactions that don't normally happen on their own to happen. Okay, just like your car doesn't normally roll uphill unless you turn it on and push the accelerator pedal and burn the gasoline in your tank to do that. A lot of things inside the cell don't happen all by themselves unless they have energy to drive them. Okay, examples would be stuff like building large molecules or making organelles. Right, that's not something that happens just like poof, just like magic. Right, it has to have energy, and that energy usually comes from ATP. Okay, all right. The ribosomes are where protein synthesis happens. So what they do is they use the information that's in the nucleus to build the proteins that the cell needs. Okay? Now that information that's in the nucleus is copied into a molecule called messenger RNA. And then that little strand of information comes out of the nucleus where the ribosomes hop onto it, and then they slide down that little strand of information, and as they read it, they're able to put together the building blocks of proteins, which are called amino acids, and make the protein that the cell needs to do the job necessary to keep the cell alive. Okay? The endoplasmic reticulum is just a membrane-enclosed series of flattened sacs, like a big, windy, twisty room. And it comes in two forms. The rough endoplasmic reticulum has ribosomes on its surface, and they make proteins that get inside the endoplasmic reticulum and eventually are going to end up moving from there to a transport vesicle, and from there to the Golgi, and then from the Golgi to a secretory vesicle where they'll be dropped off and do their job. Okay? The smooth endoplasmic reticulum doesn't have ribosomes on it, and it's where we manufacture things like lipids and steroids, and we store calcium, and we also do a lot of detoxification reactions. The Golgi is just where proteins that are delivered from the rough endoplasmic reticulum get modified, meaning that they get little chemical groups added to them that serve to help them function, but also serve to determine where they're going to go. So this is sort of like an address label that gets put on the protein, and then those proteins that have the same address label are going to be loaded into the same secretory vesicle, and then they'll be dropped off at the same location. Okay, and that could be the cell membrane, or could be an organelle, or they could end up being secreted to the outside. Right? And again, all of these processes require energy, usually in the form of ATP, in order to do their job. Lysosomes are designed to recycle material that's worn out, so those could be organelles or molecules. It fuses with the stuff that's going to be broken down, and then there's proteins inside it called enzymes that tear up whatever the lysosome takes in, and then the products that are produced, the material that's left over, can be used to build new things, or some of it can be thrown away. All right, so I like to think of the lysosome as the stomach of the cell. The cytoskeleton is just a series of proteins. They're long filaments or tubes that give the cell shape. Okay, microfilaments are strands of protein that are involved in movement of certain material, and microtubules are going to serve like pulleys or train tracks that allow organelles to move around, and they're also important when the cell gets ready to reproduce itself in a process known as mitosis, which we'll get to a little bit later. The centrioles are little rod-shaped structures that are going to manufacture microtubules and also break them down. And they also serve as the foundation for um, a structure called a cilia, which is a moving sort of hair on the surface of a cell that pushes stuff across uh, the top of the cell so it can go to another location. Or it can also serve as a part of something called a flagella, which is like a looks like a tail on a tadpole, and it beats so that the cell can paddle from one location to another. In human beings, the only cell with a flagella is the sperm cell. Microvilli are there to increase the surface area across the cell membrane so that it can be more effective at either secreting stuff, which is spitting stuff out, or moving stuff inside it and then transporting it to another location. Okay. The cilia we've discussed, right? What makes the cilia and the flagella move is the use of the energy source ATP. Okay. And that's, again, an important point, right? That energy is there to immediately do work. Okay? This is a look at the cell membrane. Stress is that the cell has to have the ability to determine what it wants to let in and what it wants to let out. And so we talked about earlier the fact that the cell membrane is what's known as semi-permeable, meaning stuff doesn't move across whenever it wants. Stuff moves across it when and where the cell wants it to. Okay, and the two types of transport that the cell can use to get what it needs or to throw away what it doesn't want are called active and passive. Okay, in passive transport, material moves across the cell membrane in a direction that it would naturally move without adding any energy to the system. Okay. Examples of passive transport include diffusion and osmosis and facilitated diffusion. And you saw examples of those in the previous video. Active transport's different. In active transport, we're trying to move material across the membrane in a direction that it wouldn't normally go. Okay? And so it has to draw on an energy source, and that's usually ATP. Sometimes it's the use of an electrical gradient that's been set up through the use of ATP. Okay? And we saw some of that in the previous video as well. So those are some of the different transport mechanisms. Okay? Active transport includes pumps and vesicle-mediated transport like endocytosis and exocytosis. The passive mechanisms, we talked about diffusion and facilitated diffusion, and we're going to see examples of those. Osmosis is just the diffusion of water, and infiltration is just the force of material against a barrier that has openings in it so that stuff smaller than the openings can go through. Okay? So let's look at these different kinds of transport. This is a look at diffusion. So you can see with diffusion that material is going to move from where it's most to least concentrated, and eventually will reach what we call an equilibrium, in which there's no more net movement of material, meaning material moving in direction A will be moving just as fast as material moving in direction B. Okay? And what ends up happening is you get an even distribution of all the stuff that's dissolved in the liquid, or in the case of a gas, dissolved in the gas. Okay? You don't have to put in any energy for this to happen. It happens automatically. Okay? Examples would be the little skunk spraying the gentleman in the panel B, or the food coloring tablet dissolving in the liquid in the panel A. Okay? Those just happen automatically. Now you can speed up diffusion by increasing the temperature, or by having a smaller thing diffuse, something with a smaller mass. Okay? Or by having a greater difference in the concentration of the thing that's diffusing from one location to another. All right? So those are just a few factors that can speed things up. You can see some examples here of diffusion in the body where it's critical, right? We need to get oxygen from the air into our blood so that the cells can use it to make enough energy to keep us alive. And we need to remove carbon dioxide from the blood so that it doesn't accumulate and produce toxic conditions that kill us. Okay? And what drives all of that is diffusion across what we call a concentration gradient. Okay? In this case, it's a gas, and the gradient is actually a pressure gradient. So where the gas will go is where there's the least pressure of that gas. Okay? So in facilitated diffusion, you've got an assistant, okay, a protein that's in the membrane that aids the movement of the material across the membrane by providing a gate or a tunnel, okay? In passive transport, you can have tunnels that are open all the time, and they're called leak channels because stuff just leaks across them in the direction of its gradient, right, from high to low concentration. Or you can have tunnels that are called gated channels, where doors are either open or shut depending on certain conditions in the cell membrane, such as the electrical charge around the gate, or whether the gate is attached to a particular chemical, or whether the gate has a certain type of physical force applied to it. Okay? And that lets material that the gate will allow through to go from where it's most to least concentrated. Okay? So in osmosis, what's happening is that water is moving from where it's most to least concentrated. All right? The idea here is that water still has to obey the same rules that something dissolved in water has to obey. And water wants to go from where it's most concentrated to where it's least concentrated. So an example of how this works is taking a cell and putting it into a solution of distilled water, right? If we did that, the water concentration outside the cell would be much greater than the water concentration inside the cell, and the water would move in so quickly that the cell would swell up and burst, and that's the situation here in the middle, okay? In a hypertonic solution, you put the cell in an extremely solute-rich solution, like really salty water, for instance, okay? So the water concentration inside the cell is greater than it is outside the cell, and the water leaves the cell more quickly than it enters, and the cell shrivels up and dies, and that's a process that's called crenation, spelled with a C. And then in an isotonic situation, you have water concentrations that are the same both inside and outside the cell, and so the movement of water in both directions is equal, and the cell continues to function. Okay, so the body works real hard to try and maintain isotonic conditions around all our cells and tissues. That's why when we give you an IV, if you can't take fluids through your mouth, right, we would never give you an IV of pure water, because you would rupture the red blood cells and your white blood cells, and they would release their contents, and that would clog all your tiny blood vessels called capillaries, and then they wouldn't get any blood, and those tissues that need those capillaries blood would die, right? So what we usually do when we give you an IV for the purposes of hydration is that we give you an isotonic IV, okay? We give you something called physiological saline, and that lets your blood volume get back up and you get to recover fluid, but you don't traumatize your cells and your tissues, okay? Now filtration is a different situation. In filtration, what happens is you've got the pressure of a liquid against some sort of a barrier that has holes in it, and anything tinier than the holes will go through from high to low pressure, okay? You see this with a syringe. You see this when you make coffee, right? From coffee grounds, or from, fresh coffee in the coffee maker. The coffee grounds don't go through the filter, but the coffee does, okay? In the body, this happens in the kidney tubules, all right? And it also happens in capillary beds all over the body, where we're able to force out a certain amount of fluid as a result of pressure, okay? And at the other end of the capillaries, where we're going to return blood back to an organ called the heart, the fluid goes in the other direction, again, down the pressure gradient. This is a look at active.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
So in active transport, what we're doing is we're moving material in a direction that it normally wouldn't move, okay? And that requires some form of energy, and the energy is usually in the form of ATP, okay? Sometimes the energy can be from an electrical gradient or a chemical gradient that's set up by another active transport pump, okay? But the idea here is that we're moving material in a direction that it doesn't move automatically, and as a result, we need to provide energy from another source. Okay? So you can see with vesicle transport, what we're doing is that we're generating a little membrane bubble, and we're using that as a chamber to move material from one location to another. In endocytosis, we grab something outside the cell and form a little membrane bubble around it and bring it inside the cell, and then that little membrane bubble could fuse with another organelle, or it could, in some cases, even walk all the way across the cell to the other side and be spit out, and that's called transcytosis. When we do the opposite, when we take material inside the cell and load it into a vesicle and spit it out, we call that exocytosis. And all of this requires energy to happen, because moving the vesicles around isn't something that occurs automatically. We have to have ATP in order for that to take place. Okay? So these little vesicles walk along the microtubules in order to drop their payload wherever they need it to be. Okay? So every cell in the body has a period of time where it does its job, and then eventually it gets to the point where it has to reproduce itself in order to replace cells that get worn out and that die. Okay? In order to do that, we have to do two things, right? We have to duplicate the genetic material in the nucleus, and then we also have to take the rest of the cell and divide that. And that's what mitosis is for. As you saw in the previous video, the result of mitosis is two cells that are identical to the parent cell that have the same DNA inside them, and as a result will look and act like the original one. So we use that for things like growth and repair. It usually obeys rules. We'll see in the next video a little bit about the cell cycle. Normally cells only grow when they're supposed to, like when other cells wear out, or when there are chemical signals in the body that tell certain cells and tissues to grow more rapidly. All right? Meiosis is different. In meiosis, we generate sex cells. In guys, those are the sperm, and in females, those are the eggs. And they have half the amount of genetic material as a cell in anywhere else in your body. And the reason for meiosis is in order to keep the number of chromosomes the same when we go from one generation to the next. Okay? Um, if we didn't do that, what would happen is the number of chromosomes in our body cells would keep doubling with every generation, and that would result in cells not functioning correctly. Okay? The cell cycle is just a sequence of events that allows the cell to know when to go into mitosis and when to basically do its job. The phases of the cell cycle include the M phase, where the cell is dividing, so it's duplicating its genetic material, and then interphase, where it's either doing its job or it's preparing for the next mitosis. Okay? Interphase is divided into G1, S, and G2. The G1 phase is essentially the cell doing its job in the body. Okay? And some cells spend their whole life in G1 phase after you're born, like nerve cells. Okay? The S phase is where we synthesize DNA in preparation for G2, where we gather all the material we need for mitosis, and then we enter the M phase, where the cell duplicates its genetic material, distributes it to two new cells, and then we have cellular reproduction. Okay? Now, normally, there are chemical signals that tell the cell when and where to obey the cell cycle, but occasionally cells end up getting changed in such a way that they don't listen to those signals and they start to divide on their own, and that's what we call cancer. Okay? And the problem with cancer is that these cells can divide very rapidly, and in many cases they can walk through the blood or through the lymph and land in other locations and then begin to divide there. We call those tumors. And essentially what the tumor cells do is they consume stuff in the blood that healthy cells need, to the point where the healthy cells can't survive. Okay? And then they end up dying, and that eventually ends up killing the patient. Okay? So the way that we attack cancer is to try to attack cell division. So we hit the patient with radiation and with chemicals that ruin cells that divide rapidly by hurting their DNA. And the result is that we end up hopefully killing the cancer cells before we hurt the patient too badly. Okay? In mitosis, the phases are prophase, metaphase, anaphase, and telophase, and the video went through what happens in each of those phases. The idea here is just to reproduce the genetic material and then to distribute it to two brand new cells. And then most of the time, that's followed by the cell dividing in half, and that's a process called cytokinesis. Okay? There are some cells that do mitosis without cytokinesis. For instance, the cells that produce the platelets in your blood do mitosis and not cytokinesis. Okay? So it's not going to be every time, but 99 times out of 100, it's always mitosis followed by cytokinesis. Often what will happen during development is that a cell will take on different characteristics that define what kind of a job it's going to do in the body. Okay? And that's a process called differentiation, and that happens mostly during human development in the womb, but it also continues after we're born. Okay? So the idea here is that you'll have a cell in the growing embryo, or the fertilized egg, or the fetus, all right, that could end up having a variety of different jobs in the adult. But what job it's actually going to do is going to depend on the location of that cell in time and space. Okay? What will happen is that those cells will see chemical signals, and those chemical signals will cause the genetic material in those cells to be modified in a way that only certain pieces of information in the nucleus can be used. The other information is still present, but it's not available for the cell to build proteins. And what that does is that changes the cell's characteristics and eventually guides it on a path to being, a nerve cell or a skin cell or a bone cell or what have you, okay? And the information that's not important to doing that job isn't used anymore, okay? And that process is called differentiation. And it happens mostly when we're developing from a fertilized egg to a baby, but it goes on in the adult too, okay? There are adult stem cells that change their characteristics as they encounter chemical signals and produce tissues that help us survive. A stem cell is just a cell that can have a variety of different fates, okay? It could be any possible tissue cell in the beginning, so they're called totipotent cells, but as they encounter chemical signals, they get their fates more and more restricted so that they can only be certain tissue types, okay? And what will happen is we'll go from a totipotent stem cell that could be anything to pluripotent stem cells that have a certain number of possible fates, and then finally you get to a differentiated cell that doesn't do any more changing and does its job in the organism as it is, okay? And that all happens as a result of changing the information that's in the nucleus, right? Making the cell capable of using less and less of it so that it turns into what the body wants it to be. Some